Welcome back, listeners, to 5 Minute Pharma, keeping you up to date with the newest drugs around, FDA approved, of course. Will and I welcome Mr. Wickham, pharmacologist, and Dr. Brown, who will be sharing must-know info on the mechanisms, trials, and studies that have driven our drug of the day, Leqembi, to its accelerated approval on the 6th of January last year.
It's uplifting to finally see some promising action around Alzheimer's disease. Don't you agree, Amelia?
It certainly is. But how exactly is Leqembi different from our existing treatment options?
Well, although the drug is a monoclonal antibody like its predecessor, Aduhelm, Leqembi or lecanemab reduces amyloid beta plaques in the brain, slowing the progression of Alzheimer's. Unlike other treatments, Leqembi has demonstrated a statistically significant impact on slowing cognitive decline, making it a breakthrough in Alzheimer's treatment. What makes the approval so intriguing is how fast it was approved and the evidence for its efficacy.
Interesting indeed, especially considering Aduhelm's approval was based only on the surrogate endpoint of amyloid beta clearance, as it failed to demonstrate any significant effect in its clinical trials. As a summary for those who may have zoned out, Leqembi works to slow cognitive decline, it's suitable for patients in early stage Alzheimer's, and it's a novel antibody targeting soluble amyloid beta plaques, and currently the most promising treatment for the disease. Now, to properly understand the mechanism of action, I'd like to call in for a pharmacological perspective. Mr. Wickham?
Leqembi is a humanised IgG1 antibody which selectively targets soluble amyloid beta protofibrils. Upon the antibody complex being formed, signals are transduced to resident macrophages to phagocytose the protofibril.
Are you able to elaborate on the pharmacodynamic properties?
The pharmacodynamic relationship is dose-dependent following a sigmoidal curve. Commonly an IC50 value is used which shows the effective concentration of Leqembi to reduce amyloid beta precipitate by half, which the IC50 value is 0.8 nanomoles for the protofibril.
Anything of note for the pharmacokinetics?
Leqembi follows first-order kinetics in a two-compartment model whereby the rate of drug clearance is proportional to drug concentration. Leqembi is delivered intravenously with steady state being achieved after 6 weeks at 10 milligrams per kilo with peak concentration of 15 milligrams per kilo. Mean distribution at steady state is 3.22 litres. Mean elimination rate is 0.434 litres a day with a half-life of 5 to 7 days, which is eliminated through proteolytic enzymes.
That's all super interesting. Dr. Brown is here now to update us on the development of this drug. Could you please tell us a little bit about the trials performed prior to Leqembi being approved?
Absolutely. As with all clinical trials, the process of developing Leqembi for commercial use involved 3 phases. What is unusual is the fact that Leqembi was approved for clinical use after the second phase. The first phase of trials was conducted in 2015, following the discovery of antibodies that could reduce amyloid beta plaques in 2004. This trial was conducted on transgenic mice models and found that Leqembi, then only known as BAN2401, was able to selectively bind to and destroy the kind of amyloid beta protofibrils that are associated with Alzheimer's disease.
That sounds promising. Was the next step to trial the drug for human use?
It certainly was. Phase 1 trials left researchers fairly confident that Leqembi could be used in humans without serious adverse effects, so they proceeded to Phase 2, human trials. An initial study was conducted with 43 participants that investigated the efficacy of different doses of Leqembi and ensured its safety for clinical use. Following this, a much larger proof of concept study was conducted with 854 subjects, which found that Leqembi was effective in reducing the presence of amyloid beta plaques in patients in the early stages of Alzheimer's disease at a dose of 10 milligrams per kilogram. At this point, the FDA noticed that Leqembi presented as a drug that could fill an urgent need where no other treatment was currently available. They granted Leqembi advanced approval on the 6th of January 2023 before Phase 3 trials commence. Though it must be said that there are some issues with Leqembi being approved so fast.
Oh really? What would that be?
One major issue is that both genders were treated as one population, and if you do split the genders into male and female, it is seen that cognitive decline was not slowed in women. In addition to this, individuals with the APOE4 gene, a genetic risk factor for Alzheimer's disease, have no response to Leqembi.
Having heard these challenges, it's clear that there's a need for future studies to be conducted. Amelia, can you elaborate on what we should do next?
Currently, the AHEAD study, which began in 2020, enrolled 1,400 participants with the risk of developing Alzheimer's disease with intent to administer Leqembi over the course of 7 years. PET scans were adopted to view these changes in amyloid beta plaques and therefore determine its ability to slow cognitive decline. Prospective studies are the way of the future. Leqembi has great potential, but due to the complex nature of the disease, it's imperative that studies look into co-administering Leqembi with supplementary drugs that target tau protein or neuroinflammation, for example. Given the design flaw in previous trials, we'd like to increase the accuracy where possible. In future trials, populations should be split into females, males, and APO4 carriers and have strict inclusion-exclusion criteria for more effective data analysis. There's also potential to develop personalised therapies for these patients with genetic predispositions, providing distinct individualised treatments for those patients.
With such an expansive future, Leqembi does look promising.
Yes, very much so. Well, thank you all for tuning in for 5 Minute Pharma. Until next time.